Okay, so thank you for joining the second AKN eTalk seminar today. My name is Lee Jae-kyung, and I'm an associate professor at University of Georgia. Today, I am so excited to introduce our seminar speaker, Dr. Han Sung from the Institute. Dr. Han earned his bachelor's degree from Gyeongbuk University, and he got his master's degree from Pohang and he then moved to the United States to complete his PhD at WashU. And following his postdoc training there, he began his tenure track position at the Salk Institute for Biological Studies in 2016, where he is now a tenured associate professor. Dr. Han's research primarily focused on the neural circuit mechanism underlying neuropeptide and receptor action. Today, he will share his most recent research program on the role of peptidergic transmission in neural circuit function and behavior. Without further ado, please join me welcoming Dr. Han.
Hello, everyone. So you guys hear my voice? Well, okay,
Yes.
great. So I would like to start by thanking the AKN eTalk organizers, Dr. Wu and Lee, and I really appreciate this great opportunity to share our recent study to the Korean neuroscientists and the Korean neuroscientists in the US as well. So uh, just wanted to correct one thing. I'm an associate professor, not yet tenured. So yeah, so Salk provide the tenure for full professor. So not there yet. So hopefully sometime soon. Okay, so yeah, as Dr. Lee introduced, I'm at the Salk Institute, and the name of my lab is a peptide biology lab, which means we study the neuropeptide. So the Salk has a rich history of the neuropeptide. Uh, like there are lots of neuropeptide discovered at Salk, and actually the neuropeptide that I'm going to talk today is also discovered at Salk. So before going through today's topic, I just wanted to um, introduce what we are doing briefly. So yeah, as I mentioned, so the main theme of the lab is the neuropeptide research. And we first focused on the peptidergic circuits that mediate negative emotions like fear, anxiety, and pain, and panic. So those negative emotions are encoded by many peptidergic circuits. So we have been dissecting. We've been interested in this topic, and we've been dissecting this neural circuits in the context of the neuropeptide. And the second theme of my lab is opioid. So the opioid is huge problem in the United States right now. It's called opioid crisis because uh, almost like 100 people die per day just by the opioid overdose, which is higher number than the people die from car accidents. So this is a huge socioeconomic problem, but the cure for opioid overdose is not being available yet. So we decided to, when I started my lab 8 years ago at Salk, we decided to uh, understand uh, opioid-induced behavior, physiological and emotional changes. The reason why opioid is linked to the neuropeptide is because uh, so the endogenous opioids are actually neuropeptide. For example, I think many of you guys heard about the endorphin. Endorphin is actually originally discovered at Salk Institute. So the endorphin or enkephalin, dynorphin, all these endogenous opioids are the neuropeptide. So we are now trying to understand opioid actions and then dissect specific peptidergic circuits that mediate opioid-induced changes using the animal as mice as a model system. And as we study the negative emotion and opioid action, we found some common features um, which is actually, I just define it as an emotion physiology interaction, but specifically the breathing. Emotion and breathing are interacting each other. As we study these two topics, we found that negative emotion greatly contribute to changes in breathing, but at the same time, opioid signaling can contribute uh, breathing regulation. So that's actually the primary reason why people die by opioid overdose is because of the depression of the breathing system by opioid. So people die by the opioid through the breathing suppression. So we decided to study how neuropeptidagic, the endogenous opioid circuit, contribute the changes in breathing and how the emotion actually contribute changes in breathing. So we uh, actually, so next month there will be a new paper published from the lab, from my lab, showing how volitional uh, so volitional thought can control the breathing. So, but this time I'm not going to focus on that. And then lastly, as we study these topics, we just realize that there are lack of the tools for specifically dissecting the peptidagic transmission in animal. Unlike other transmitter systems like monoaminergic or fast transmitter system, the tools for dissecting peptidagic transmission, for example, a sensor for detecting the release of the neuropeptide or silencer for uh, suppressing the peptidagic transmission was not available. So we decided to develop that tool. So today's topic is more about this. Half of the talk today is going to be in this category. So the tool development. But the next half, I'm just going to share the novel discovery that's not been published yet using these new tools. So the topic today is about the synaptic transmission. So you guys, so we all know that neurons, the brains works through the network of the neurons, and then each neurons communicate one another uh, uh, through the synaptic transmission. There are two types of synapse that where the synaptic transmission happens. One is a chemical synapse, the other is electrical synapse. But I'm going to focus on the chemical synapse because the topic today is about the transmitter. So we all know that the neurotransmitter is the messenger of the neurons for relaying the information from the to the next the neighboring neurons. So and then there are many types of the neurotransmitters. For example, glutamate, GABA, glycine, or biogenic amines, or acetylcholine. All these act as a neurotransmitter, but there are another types of the chemicals that contribute the neurotransmission is called the neuromodulators. For example, norepinephrine, serotonin, dopamine, and neuropeptide, today's main topic. So the difference between neurotransmitter and neuromodulator, there are many differences, but the main difference is the neurotransmitter acts really fast through its downstream ion channels, but the neuromodulators usually works through the GPCRs, the G protein coupled receptor, so that it's kind of slower than the neurotransmitter. But I mean, this summary is easily found in the textbook, neuroscience textbook, but I don't think this is really the correct uh, classification of the chemicals that use for the transmission, because sometimes the glutamate, which defined as a neurotransmitter, can act as a neuromodulator through the GPCRs. It's called motabotropic glutamate receptor. GABA, the same, and acetylcholine, same. The muscarinic acetylcholine receptor is actually G protein coupled receptor. So these neurotransmitter can act as a neuromodulator. And also, opposite case is also possible. So monoamines like dopamine, some some dopamine neurons only packages dopamine as a transmitter. So it is a sole transmitter for neurons to use to transmission. So some dopaminergic neurons definitely use dopamine as a main neurotransmitter. So I would like to just redefine this neurotransmitter and neuromodulator with different terms, at least in my talk today, which is so the neurotransmitter, I would say fast transmitter, and the neuromodulator, I would like to call it as a slow transmitter. So throughout the talk, I'm just gonna define a conventional neurotransmitter as fast transmitter and conventional neuromodulator as slow transmitter. So these two different types of the transmitters are packaged in axon terminal inside specific class of the vesicles. For example, I mean this everyone knows this. The fast transmitter like glutamate or GABA are packaged in the synaptic vesicles, the clear vesicle. This is the EM image of the synaptic terminal that shows distinct classes of the vesicle that contains different types of transmitters. Here you see clear vesicle, clear small vesicles. These are the synaptic vesicle, and there are actually distinct vesicles, which is larger than the synaptic vesicle, and the core is stained with a dark signal. So without any staining. So this characteristic called define the name of this specific class of the vesicle as a large dense core vesicle. So these are called large dense core vesicles, and these large dense core vesicles specifically packages the neuropeptide, not other transmitters. So take-home message here is different classes of the transmitters are packaged in different vesicles, and release property of these different vesicles actually distinct. For example, synaptic vesicle can be released even by single action potential. However, large dense core vesicles cannot be released by single action potential, but it requires trains of high frequency action potentials. So this is basically the introduction. And our central question that in my lab is, can neuropeptides act as a main transmitter? So this is not just our question. This is actually the question from the founder of the my lab. So the name of the my lab is Peptide Biology Lab. And the founder of the Peptide Biology Lab is Roger Guillemin, who is a Nobel laureate. He got Nobel Prize at 1977. He founded the Peptide Biology Lab, and then several investigators actually succeeded. And then I'm currently a member of the Peptide Biology Lab. And the Peptide Biology Lab as well discovered lots of uh, uh, famous neuropeptide like CRF, for example, corticotropin releasing factor, or uh, somatostatin. Somatostatin is not just a marker of the interneuron; it's actually the neuropeptide. And another famous neuropeptide is endorphin, the beta endorphin. Yeah. So these are, among other neuropeptides, these are discovered at Salk, and Salk has a rich history of the peptide uh, neuropeptide research. And at his Nobel lecture, what Rose defined is these peptides at that time, these peptides expressed in neurons uh, that we called hormones earlier do not fit the definition of the hormone anymore. They seem to be a candidate for the definition of neurotransmitters. So at his Nobel lecture 1977, Roger Guillemin, he already proposed that neuropeptide can act as a neurotransmitter, but there's an idea about it, but nobody actually visualized and functionally tested whether a neuropeptide can act as the primary transmitter. And then later, like 8 or 9 years later, at his last public lecture, Roger uh, claimed more bold statement here, since there are so many of the peptides with different specific structures and so few of the classical transmitters, I personally prefer the concept proposing that the neurotransmitter modulate the secretion of the peptide. So in this lecture, he claimed that actually neuropeptide can act as a neurotransmitter, but the conventional neurotransmitters like glutamate GABA actually uh, modulate the function of the neuropeptide. So I I should confess that I have really a bias on the neuropeptide research, but at least certain neural circuits, it's definitely possible. So we decided to address this question by developing new tools. So the circuit that we are interested in is a brainstem circuit that expresses a neuropeptide called CGRP, calcitonin gene-related peptide. This CGRP is also discovered at the Salk Institute at 1983. And this brainstem CGRP neurons are well known for relaying effective pain information to the amygdala during Pavlovian field learning. So this circuit acts as an unconditioned stimulus circuit. And then as a postdoc in Dr. Richard Parmaiter's lab at University of Washington, I dissected this circuit, and I found that the neurons in the parabrachial nucleus that express CGRP actually relay the pain information during the fear conditioning so that they relay push-up information during the conditioning. Uh, yeah, but the question I had was, after finishing this project, whether these CGRP neurons using um, glutamate as main transmitter or CGRP as a neurotransmitter. So that was my question after I moved my to the Salk Institute, because the CGRP neuron, all CGRP neurons co-express vesicular glutamate transporter type 2, indicating that these are the glutamatergic neurons. But um, my circuit dissection performed at the Richard Parmaiter's lab was just activating or inhibiting the activity of the CGRP neurons. So we don't know whether CGRP neurons use glutamate as a main transmitter or the CGRP as main transmitter. So after opening my lab, I decided to study this topic. Which one, glutamate or neuropeptide, which one is the main transmitter to encode fear during a Pavlovian fear learning? But in order to test this idea, we need a tools. So because the easiest experiment to test this idea is just monitoring the release of the glutamate or neuropeptide when the animal receives push-up. And then another experiment that's required is uh, silencing glutamate specifically or neuropeptide specifically and see what happens during the fear conditioning. So those two critical experiments required new tools for monitoring or silencing the release of different transmitters. Currently, the glutamatergic transmission can be easily monitored by lots of sensors, but the neuropeptide sensor was just recently published by Yulong Li lab at China last year, but that was the postsynaptic sensor, not presynaptic sensor. So it can indirectly monitor the release of the neuropeptide. Um, and for the silencing each transmitter, the glutamate silencing tools are available, which is a CRISPR-Cas9 approach to knock out the VGLU2 gene. But the peptidogic transmission silencer was not available when we studied this circuit. So we decided to develop a sensor or silencer for monitoring or silencing the release of the neuropeptide. Okay, so developing neuropeptide sensor, um, we utilize the specific protein, which is a fluorescent protein called fluorine. Fluorine is the mutated version of the GLP, the green fluorescent protein. But unlike the green fluorescent protein, fluorine is sensitive to pH. At low pH, fluorine does not generate the fluorescent signal, but at neutral pH, fluorine generates the green fluorescent signal. So by detecting the intensity of the green fluorescence, we can actually detect the pH changes. And then why we use this fluorine construct as a sensor? Because the dense core vesicle and synaptic vesicle, the luminal side of their vesicle, the inside the vesicle, the pH is acidic, about 5 or 5.5. So before releasing the before the vascular release, this inside luminal pH is acidic, so there's no fluorescent signal. If we target this fluorine inside the luminal side of the vesicle, each vesicles, However, during the vesicular release event, this pH can be changed to 7.3, the neutral pH, because the synaptic cleft pH is 7.3. So by just monitoring the intensity of the fluorescent signal, we can detect whether each vesicles release or not. So in order to find a specific marker for larger than square vesicle for peptidogic transmission, we focus on the enzyme critically involved in neuropeptide maturation. So among dozens of the candidates that's specifically known to be specifically expressing in the large and small vesicle, we narrow down these two proteins, membrane proteins. One is called peptidoglycine alpha amylating monoxidase, in short called PAM, and the second was the cytochrome B561. Previous research already confirmed that these two membrane proteins are specifically located in the larger than square vesicle. So we genetically targeted loop of luminal side of each transmembrane protein uh, to make a fusion construct that makes fluorine, the synaptic superecliptic fluorine, localized inside the luminal side of the larger than square vesicle. So by doing it, we can detect fluorescent changes during the transmission event of the larger than spore vesicle. So actually, I originally just tried to explain all these things, but I decided not to share the characterization of the data for the characterization of this sensor. Instead, I decided to focus more on the new unpublished result that I'm going to share today in the later slides. So for the validation or characterization of the presynaptic neuropeptide sensor, please go to the PubMed and find a recently published paper from the lab and read the manuscript. So we successfully validated in expression cell line and also in the brain slice and in awake behaving animal. So we confirmed that our approach works really well as a marker of the larger than square vesicle release. Okay. So and then so make it simple. So we generate this construct as a larger than square vesicle sensor, and we also targeted this pH sensor to synaptophysin so that we can regenerate the synaptic vesicle sensor. So the animals expressing this synaptic vesicle sensor, we can detect the release of the glutamate or the fast transmitter package in the synaptic vesicle. Or if we express this sensor in animals, then we can actually detect the large dense core vesicle release, which is a peptidogic transmission. And then the fluorescent change can be detected by bulk fluorescent detection method called fibrophotometry in awake behaving animal. So here's the result. So we express in different group of animal, we express either dense core vesicle sensor or synaptic vesicle sensor and deliver the foot shock. So we just basically deliver the same aversive sensory information during the fear conditioning. And interestingly, what we've discovered is during the foot shock event, the large dense core vesicle sensor expressing animal has a time lock increase of the fluorescent signal, but the synaptic vesicle sensor expressing animal, the foot shock event didn't increase the fluorescent signal. Rather, actually, there's a slight decreases. Uh, so we didn't actually believe this first, but throughout the validation, this is really the physiological event, and I'm going to cover this in a later slides, why this decrease was observed. But anyways, during these results indicate that the foot shock triggered the release of the neuropeptide, but not the glutamate in at least in the CGRP neuron that projects to the central nucleus of the amygdala. Okay, so but this is just a correlation analysis. We just observed that during the fear conditioning, the foot shock triggered the release of the larger than square vesicle, not the synaptic vesicle. But that doesn't mean that the neuropeptide is really necessary for encoding uh, aversive information during the fear conditioning. So in order to test that, we need a loss of function study by removing the neuropeptide signaling to see what happens, or removing the glutamatergic signaling, what would happens. If the glutamate is the main transmitter, if we remove the glutamate, maybe we can substantially reduce the fear acquisition. But if the neuropeptide is the main transmitter, if we remove the neuropeptide, then we can actually block the fear acquisition. So, but there's no such tool to silence the peptidogic transmission. So we decided to develop a neuropeptide silencer. So this is actually from my idea, but my postdoc Dong-il Kim actually made this construct. So what my idea was, there's actually the enzyme called the neutral endopeptidase, which is endogenously, this neutral endopeptidase is expressed in the outer surface of the plasma membrane. So this is the natural inactivation mechanism of the peptidogic transmission. Once the neuropeptide is released, the outside plasma membrane, this neutral endopeptidase located in the outside plasma membrane just digests these neuropeptide. And this neutral endopeptidase is specific for neuropeptide, not other proteins. So it is called encapsulinase because it also digests encapsulin, but it doesn't digest other proteins. So this is a neuropeptide specific endopeptidase. But we genetically engineered catalytic subunit of this NEP and then targeted this inside the vesicle, the large dense vesicle, so that this NEP digests most of the neuropeptide packaged inside the large densicle vesicle. So we can silence the peptidogic transmission without changing the glutamatergic transmission. Again, we perform all these characterization and validation of this presynaptic neuropeptide silencer in this the recently published paper. So I decided not to cover all those characterization and validation results because I wanted to focus more on the biology that we discovered using this sensor and silencer. So using this silencer, we tested whether peptidogic transmission is really critically involved in fear conditioning. So we express this the, the neuropeptide silencer and then perform the fear conditioning. And as you see here, in Q-dependent and context-dependent conditioning, you see that there's a substantial reduction of the freezing behavior in animals that express a silencer for peptidogic transmission compared to M-cherry control. Um, but this is just a silencer for all the neuropeptide that is expressed in the CGRP neuron. So during the review process, one reviewer actually asked a question: Is this neuropeptide silencer block the freezing behavior by inactivating CGRP signaling, or other neuropeptides are also involved in this fear learning? And then during the revision experiment, we wanted to test whether CGRP in this CGRP brainstem CGRP neuron is the main neuropeptide that transmit aversive sensory information, or other neuropeptides are also involved in this transmission. So in order to silence the CGRP signaling, we knock out the CGRP receptor. The reason is because there are two genes that encode CGRP. One is the CGRP alpha, which is called CALCA, CALCA. But there's actually another gene that encodes the same neuropeptide called CGRP beta. So that is called CARC-B. So there's no such tool to silence CGRP alpha and CGRP beta. So we just decided to knock out the CGRP receptor in the downstream neuron to test whether CGRP signaling is involved in relaying the push-up information during fear conditioning. And then we found that knocking out the CGRP receptor using the, the, the free system, we didn't see any freezing changes, indicating that the CGRP signaling is not important for uh, fear learning. So which was quite surprising to us because we really believe that the CGRP is the main transmitter for this pathway. But previous research showed that injecting the CGRP directly into the amygdala can induce strong freezing behavior. So that means CGRP can encode uh, push-up information during fear learning. However, it is not necessary. Means other signaling molecules can take over the role of the CGRP when the CGRP is removed. So there's a compensatory mechanism. That is our understanding because the genetic knockout of the CGRP doesn't change any danger-related motivational response. There's a recent publication, and the paper published this year, um, uh, knocking out the CGRP and substance P, which is another neuropeptide known to relay the pain information. So double knockout of the substance P and the CGRP doesn't change the pain perception behavior. So these previous research in line with our current uh, data show that the CGRP is somehow encoding the pain information during the fear conditioning. But if we remove the CGRP, it is still okay because other signaling molecule can compensate the role of the CGRP. That is our current understanding because previous research show that this is axon terminal. You see, again, these clear vesicles are synaptic vesicle that packages uh, uh, the neurotransmitter like glutamate. And here there's large dense core vesicles. And this group, sorry, I actually forgot to add the reference here, but this was published in 1987, showing that single large dense core vesicle that's known to package the neuropeptide can package it multiple neuropeptide, not just single neuropeptide. For example, here CGRP and substance P are co-packaged in this single large dense core vesicle, which means during the peptidergic transmission event, not only the CGRP is released, but other neuropeptides are simultaneously released. And then CGRP neuron in the parabrachial nucleus that we study express dozens of the neuropeptide. And then top three candidates that express the most highly expressing in the CGRP neuron in the parabrachial nucleus are CGRP and substance P and PACAP, pituitary adenylate cyclase activating peptide. So those three are the most abundant neuropeptide expressed in parabrachial CGRP neuron. And all these three neuropeptides are co-packaged inside in single large dense core vesicle. So the fuchsia can trigger the release of the large dense core vesicle, and then during this transmission event, multiple neuropeptides can co-release. So removing single neuropeptide, for example, CGRP, is not sufficient to block peptidergic transmission. That was our hypothesis. So in order to test that idea, we just simply inject antagonists of the receptors for those three neuropeptides, which are expressed in the central amygdala. So we just implanted the cannula and then perfused the cocktails of the antagonists for those three neuropeptide receptors, CGRP receptor antagonist, and PACAP receptor antagonist, and substance P receptor antagonist. And then we compare CGRP antagonist alone. And as you see here, there's a substantial reduction of the freezing behavior in mice that treated with different antagonists for different neuropeptide receptor. But the CGRP receptor antagonism is not sufficient to block freezing behavior. Not only the freezing behavior, the pain score. So we performed with the same animal, we perform the formalin assay, and then just score the behavior. And as you see here, oh, actually, this is the data published previously showing that knocking out the entire CGRP population, activity of the CGRP neuron with HM4DI, we observed a substantial reduction of the leaking behavior, indicating uh, pain perception was substantially reduced by inhibiting the CGRP neuron. But this is just changing the activity of these neurons. So um, we treated the cocktail of those three neuropeptide receptor antagonists and performed the same formalin leaking behavior assay. And as you see here, compared to the CGRP antagonist, CGRP receptor antagonist alone, the cocktail of the antagonists substantially reduced the leaking behavior, indicating that the pain perception is also, the multiple neuropeptides are also contributing to the pain transmission. And then CGRP alone is not sufficient to relay the pain information. Blocking the CGRP alone is not sufficient to block the pain transmission. So that was our conclusion. But this doesn't mean that neuropeptide is the main transmitter, because in order to claim the neuropeptide is the main transmitter, we need to test the involvement of the glutamate during the field conditioning. Otherwise, we cannot say that. So what about glutamate during the field conditioning? Interestingly, we didn't test but my postdoc lab, Dr. Richard Parmiter's lab, um, actually, after I moved to Stoke, Richard had the same idea as I had. So he was also interested in um, identifying the main transmitter during the field conditioning in this CGRP, parabrachial CGRP population, because he also knew that the parabrachial CGRP neurons also are glutamatergic neurons. But the difference is, the Richard's idea is that the neuropeptide is just a marker, and he thought that the glutamatergic transmission is actually the main, glutamate is the main transmitter for this circuit during the field learning. So Sagan Park from KAIS, he joined Richard's lab after me, and then he actually took this project. So Sagan dissected the same circuit, and then he silenced the glutamatergic transmission by expressing uh, uh, the CRISPR-Cas9 tools to silence the VGLUT2 gene, so that they confirmed that the VGLUT2 protein was substantially reduced in exon terminal of the CGRP neuron. And they, using the electrophysiology, they confirmed that the glutamatergic transmission was substantially reduced. But there's some residual left, so they also performed the genetic knockout, not the viral approach. But either viral approach or genetic knockout, they didn't see any freezing changes when they performed the field conditioning with animals expressed this CRISPR-Cas9 system. So they concluded that glutamatergic transmission is not important for fear, the relaying the aversive sensory information during the field conditioning in the parabrachial CGRP. So in conclusion, um, I hope I provide enough evidence that neuropeptides, not glutamate, are the primary transmitters for peptidologic affective pain circuit in brainstem area. And also we uh, found that not just single neuropeptide, CGRP, but multiple neuropeptides are required for the peptidologic affective pain circuit in the brainstem. These data suggest that blocking one peptidologic neuropeptide receptor as a therapeutic intervention, for example, like a pain. So actually, the substance P receptor, which is a TEC-R1, was a really promising target for the developing analgesics. However, the clinical trial failed. Human clinical trial failed. But we think that the reason why it's failed is because although substance P is really important neuropeptide for transmission of the pain information from the spinal cord to the brain, so the neurons that relay those signals contains multiple neuropeptides. So blocking, antagonizing one neuropeptide, in this case, substance P receptor, is not sufficient to efficiently block the pain transmission because um, other other neuropeptides are co-released, and then the the downstream G protein coupled receptors for those neuropeptides is still intact, so that in order to efficiently block pain transmission, I think we need to target multiple GPCRs. That is, I think, important information that we discovered. Okay, so then the, in order to claim neuropeptide is acting as really the main transmitter, actually, one missing information that actually we didn't test is whether peptidergic transmission is sufficient to drive the downstream neurons to fire action potential. So the neurotransmission, classical neurotransmission, the presynaptic neurons release neurotransmitter, and the downstream neurons uh, fire action potential in response to this synaptic transmission. So in order for the neuropeptides to act as a neurotransmitter, the neuropeptide alone without the glutamate should be able to generate action potential in downstream postsynaptic neurons. But we didn't test that idea. But luckily, the reviewers didn't ask that question. And then recently, there's a paper published in Neuron from Mark Enderman's lab. They actually provided convincing evidence that transient cyclic AMP production drives rapid and sustained spiking in brainstem parabrachial nucleus to suppress feeding. So what they found is they just they using artificial tools that increase the cyclic AMP signaling dramatically. They found that just transient increase of the cyclic AMP is sufficient to drive the downstream neuron fires action potential. So that means the neuropeptide transmission can be sufficient transmitter for generating action potential in the downstream neuron. But I don't want to overstate this observation. This cannot be applied to all the neurons, only the subcortical neurons that express a certain neuropeptide. So that is my conclusion. So in most cases, glutamate can act as a main neurotransmitter, but certain peptidergic neurons in subcortical area may use the neuropeptide as the main transmitter. And one example is the parabrachial CGRP neuron during Pavlovian fear conditioning. Uh, so that is the first part of the talk, but I already spent about like 40 minutes. So I think I only have 10 minutes, but I think this is really exciting project. So we actually found that the same CGRP neuron. So then maybe you guys may ask a question. Then what is the role of the glutamate in this CGRP neuron in the parabrachial neurons? So we actually proposed a mechanism that a single neuropeptide neuron can release neuropeptide and glutamate at different circumstances by changing the firing rate. So I would just name it as a frequency-dependent differential transmitter release. So multiple transmitter neurons, in our cases, the neuropeptide and glutamate are co-expressed in the same axon terminal, as you see previously in EM image. And then synaptic vesicle contains glutamate, and the larger than square vesicles contains neuropeptide. So prevailing hypothesis, and then well accepted idea, which is like a dogma, is that low frequency firing only releases a synaptic vesicle. As I mentioned previously, synaptic vesicle can be released by single action potential. However, the large dense core vesicle requires high frequency train of the high frequency action potential. So people's idea is that when the neurons that co-express neuropeptide and the glutamate, the low frequency firing trigger the release of the synaptic vesicle. So the glutamate is main transmitter. But as neurons increase the firing rate, the synaptic vesicle and the large network vesicles co-released. So so this throughout this co-release, the main transmitter, so the glutamate can act as a main transmitter, and the neuropeptide can act as a modulator so that it helps the transmission of the glutamatergic transmission, either increasing the gain or inhibiting the decreasing the gain. So this is the prevailing idea, but nobody ever monitored the co-transmission in awake behaving animal. So it's not been performed in vivo whether this co-transmission event happens. So we wanted to test it. But interestingly, the brain area that we focus on, the parabrachial nucleus, previously known as the hedonic center. So here, Kent Berry's lab at the University of Michigan, they proposed that parabrachial nucleus is actually the one of the hedonic center that in, that actually encode pleasure response in animal and in human. However, a recent study also showed that the parabrachial nucleus is relaying nociceptive information to the amygdala and other brain areas. So single brain area can encode both pain and pleasure. But this is not the only brain area that has those dual functions. For example, hypothalamic regions, like David Anderson lab at Caltech, they show that aggressive behavior and the mating behavior, which is actually opposite of in terms of the valence, uh, those uh, those are mediated by the single population of neurons that express estrogen receptor type 1. So single uh, estrogen receptor neuron in the BMHBL of the hypothalamus can encode both mating behavior and the aggression behavior. So how this can happen? So the prevailing hypothesis is that there are actually subpopulation of the neurons. In our cases, in the parabrachial nucleus, there are subpopulation of the neurons that encode positive valence for hedonic pleasure encoding, and the other subpopulation that encode pain information. So the same brain area can uh, relay, encode both pain and pleasure. But there's another hypothesis that we actually wanted to test, which is single homogeneous population that express both neuropeptide and the glutamate can actually release different transmitter in response to different stimulus. But nobody tests that idea. So we wanted to test with our tools and for monitoring and silencing the neuropeptide. So first, we wanted to test so we just expressed the glutamate sensor and the neuropeptide sensor and delivered aversive sensory stimulus, in this case, foot shot. And also to the same group of animal, we delivered the sucrose, which is a positive valence. So interestingly, the neuropeptide release in response to those two opposing sensory signal dramatically changes the peptidergic transmission, differentially released neuropeptide. Neuropeptide was differentially released by those two opposing sensory stimulus. The aversive sensory stimulus increased the neuropeptide release. However, appetitive sensory stimulus didn't evoke the release of the neuropeptide. But oppositely, the glutamate release, aversive sensory stimulus decreased the glutamate release, but appetitive sensory stimulus increased the glutamate release. So let me, yeah. So we were quite puzzling why this happened, and then we had a hypothesis that maybe the same CGRP neuronal population uh, activated by aversive and appetitive stimuli, and then fire differentially so that uh, uh, different sensory stimulus actually trigger the release of the different vesicles. The reason why we had that idea is because it's been already known that large dense core vesicle requires high transient action potential but the glutamatergic vesicle requires only single action potential. And then, yeah, so we just tested that idea by artificially stimulating the parabrachial neurons and then monitor the release of neuropeptide and glutamate in awake behaving animal. So as you see here, and as we expected, this is a peptidergic transmission, the sensor for the neuropeptide release. Low frequency stimulation didn't increase the neuropeptide release, release of the neuropeptide, but as the stimulation frequency increases, there's a robust increase of the peptidergic transmission. But obviously, the glutamatergic transmission was increased at a low frequency, but as we increase the firing frequency, the fluorescence signal was actually decreased even from the baseline. So we originally thought that this was kind of artifact, but we wanted to test whether this is really a physiological, really relevant observation. So to test that, we used another sensor. So in this case, um, we just monitor the release of the synaptic vesicle. So this is not really the monitoring the release of the glutamate, but just the transmission event of the synaptic vesicle. But in this case, using different sensor uh, developed by Lauren Luger's lab previously at Janelia Farm at HHMI. So this iGlu is the postsynaptic glutamate sensor. So this is a fluorescent protein in the presence of the glutamate. This changes the conformation and it increases intensity of the fluorescence in the presence of the glutamate. So the increase of fluorescence signal means there's an increase of the concentration of the glutamate. And then we express these sensors in postsynaptic neuron, in our cases, central nucleus of the amygdala. And then we artificially stimulated the parabrachial neurons. And as you see here, there is a frequency-dependent biphasic release event. At low frequency, the glutamate concentration was increased robustly. However, as we further increase the firing frequency, the glutamatergic transmission again decreases even from the baseline. So we interpret that either in the endocytosis was increased or the spontaneous activity of the glutamatergic transmissions also decreased by high frequency action potential somehow with unknown mechanism. Anyways, we confirmed that at low frequency, synaptic vesicle release is the prominent, but as at high frequency, the synaptic vesicle release was decreased. Rather, larger than score vesicle release was the primary event. So we tested this observation with sliced electrophysiology because sliced electrophysiology is the direct measurement of the glutamatergic transmission. And also we can measure the peptidergic transmission in a slice electrophysiological recording. So since I'm running out of time, I'm just gonna show the data without the further explanation, showing that the downstream neuron in the amygdala area, and then optogenetically shining the light in the axon terminal coming from the parabrachial CGRP neuron, shined optogenetically, and then, yeah, so shine the optogenetic simulation with 4 hertz or 40 hertz in the vortex recording, and we observed there's a robust postsynaptic current in response to the 4 hertz simulation. However, 40 hertz simulation actually substantially suppressed the optogenetically evoked postsynaptic current. But the same neuron, but switch the clamping mode from voltage clamp to the current clamping recording. In this case, we monitor the release of the neuropeptide by monitoring the slow action potential changes. This slow action potential change is the signature of the neuropeptidergic transmission. So neuropeptidergic transmission is only evident at high frequency stimulation, not low frequency stimulation. These data indicate that low frequency stimulation glutamatergic transmission is the main transmission event, and high frequency stimulation, not the glutamatergic, but the peptidergic transmission is the main transmission event. Okay, so then if this is really the case, we need to show that single CGRP neuron fires differentially in response to different sensory stimulus. For example, like tail pinch or sucrose, the firing rate should be different in single CGRP neuron. So we actually, Professor Yongsang Jo from Korean University actually collaborated with us, and then his lab, he recorded uh, the the firing rate of individual CGRP neuron in the parabrachial nucleus using optotetrode optotagging experiment. And what his lab found that single CGRP neuron fires in response to the sucrose and aversive pinch. This sucrose is appetitive and hedonic sensory stimulus, but the firing rate is different. The sucrose fires about like less than 10 hertz, and then aversive pinch, which trigger the firing of the same single CGRP neuron to fire about 40 hertz. So, which is perfectly in line with our observation. So we observed that uh, the sucrose triggered the release of the synaptic vesicle, the glutamatergic transmission, um, because sucrose triggers action potential firing in this CGRP neuron with low frequency. And the low frequency firing only triggered the release of the synaptic vesicle. Oppositely, neuropeptides release in response to the aversive sensory stimulus. And then for the neuropeptidergic vesicle to be released, it requires high frequency action potential. So single neuron, EGRP neuron, in response to the pinch, fires about like 40 hertz. So single CGRP neuron can fire low frequency, high frequency in response to different quality of the sensory stimulus, and then they release different transmitters. But these are all correlative data. In order to claim different transmitter actually is mediating and encoding distinct information, we need to do the loss of function study. So we need so removing the glutamate or removing the neuropeptide. Then, if our hypothesis is correct, removing the glutamate only changes the appetitive behavior, and removing the neuropeptide without changing the appetitive behavior, but it only changes the aversive behavior. So that really happened. Oh, before then, easier test for the gain of functions experiment. If really the single CGRP population encode both the appetitive and aversive information, we can simply test by optogenetically activating these neurons with different frequencies. So we express channel rhodopsin and the CGRP neuron in the parabrachial nucleus, and then shine the light with 4 frequency and 40 hertz, 4 hertz frequency and 40 hertz frequency, and then perform the real-time place preference test. And then as you see here, 40 hertz, as multiple labs already confirmed, 40 hertz stimulation is super aversive, so the animals avoid the chamber that paired with 40 hertz stimulation. However, same group of the animal, 4 hertz stimulation can substantially increase the time in chamber, which means single peptidylogic population, depending on the optogenetic frequency, you can encode both positive and negative valence. And then we performed the loss of function study, but it's been already past an hour. So let me just summarize that knocking out silencing peptidylogic transmission actually block aversive responses, and then silencing glutamatergic transmission blocks appetitive response. That's what we observed. So to summarize, single CGRP neurons release different transmitter in response to different firing rate. And then we show that low frequency firing triggers the synaptic vesicle release, so the glutamate release, with minimal LDCB release. And high frequency firing evokes neuropeptide release with minimal synaptic vesicle release. So together with this observation, we found that single peptidogic neurons that express CGRP can encode distinct information via frequency-dependent differential transmitter release. Okay, sorry that I just late for about 10 minutes, but with that, I wanted to thank my lab members, especially the co-first authors, Dongil, Jino, Seokjae, and Xiyoung, and Dr. Jo, who are mainly involved in this second part of the project that I shared today. Thank you very much for your attention, and I know it's already been 6 p.m., but if time allows, I may take a few questions. Thank you very much.
Thank you, Dr. Han, for sharing your exciting research program, including the unpublished data. So, well, if we're gonna get some, question from the audience or in the
So
chatting
I
box.
think to facilitate the discussion, why don't we use Korean? And for those
Yeah,
who
sure.
use Korean as second language or native speaker in English, you can also ask a question in English, so I can respond in English. But for Korean, I can use Korean language. 감사합니다.